Hello, everyone. This is Kelly Eversole, the Executive Director of the International Wheat Genome Sequencing Consortium. And I'm joined today by Ricardo Ramirez-Gonzalez from the John Innes Center, who's going to give us a webinar about genome-specific primer design. But before we start that, I'll give a quick overview of the International Wheat Genome Sequencing Consortium, and then we'll move on very rapidly. I assume I will be able to. Yeah. So the IWGSC has more than 3,000 members in 70 countries. We have 8 sponsors and 826 institutes and companies that are involved in our activities. And we would not be able to have the webinar series without the generous support of our sponsors. So I'd like to thank them up front for their support and for their leadership in helping us develop genomic tools and resources for the community. And for our vision in what we call IWGSC Vision 2.0 is to enhance breeding through an increased understanding of the molecular basis of traits and their allelic diversity. Our activities include continuing our efforts and expanding our efforts with Arbor Biosciences. We hope yet in 2020 or early 2021 to publish RefSec version 2.1 and annotation version 2.1. We are still trying to get funding for the IWGSC Diversity Project, and we're also trying to make arrangements for the pre-publication release of genome sequences for elite wheat varieties and other genomic resources. And of course, fortunately for us, we were prepared as of the beginning of this year to actually go forward with our webinar series. Today, our next webinar, excuse me, is in November, and it will be with Philippa Burrell, who will talk about decoding the polyploid wheat genome using gene networks. And you can register through this link or through the regular emails that you will receive from the IWGSC. So just to remind you how the webinar works, it is being recorded and will be posted on the IWGSC YouTube channel in a few days. You can subscribe to the channel so you won't miss the upload. The presentation will be followed by a Q&A. And if you want to submit questions, please submit them using the Q&A panel. Do not use the chat panel. You can use the chat panel, however, to talk with fellow attendees and organizers. And already you should be able to download both this presentation that I'm giving and Ricardo's presentation in the handout link. So at this point, I'm going to turn it over to Ricardo, and he is going to give us a webinar on genome-specific primer design with PolyMarker. So Ricardo, thank you for being with us today, and I'll turn it over to you and come back at the end.
Thank you, Kaylee, and thank you everyone for attending. So yes, I'm Ricardo, and I'm going to be talking about genome-specific primer designs with PolyMarker. And a lot of it's not going to be only about PolyMarker, but about the ideas behind it. First of all, I'm not a biologist. I'm a computer scientist that I've been working on biology for over 10 years now. So I sit nicely in between. This is an old picture of me, of what people may think that a bioinformatician is. You wear a lab coat of biologist and work with high performance computers. Anyway, the first thing is to know why are we doing genetic markers. So I started this project during my PhD because we had a typical example of with research where you have a population of lines with a phenotype. In this case, we have a resistant line and a susceptible line to yellow rust. And as part of the problem, we wanted to have a set of markers that will be diagnostic to the resistance and a genetic map. I remember on those early days, my supervisor said, "Oh, you can do this manually. You can have you find your SNPs and you can design your primers manually." But it is a very labor-intensive job, and I thought, like, I'm gonna make mistake. I'm careful, but after doing 10, 20, I will go crazy. So I decided to automate it using my computer science background, and that's how PolyMarker was born. So one of the initial idea was to make SNP markers diagnostic for this particular genetic map. But of course, the markers can also be used to find out if the lines that you are growing are the lines that you think they are. So you can genotype them, or you can also use them on breeding programs for marker assisted selection. In particular, I started developing PolyMarker for KASP markers because it's the technology that we have in the lab and that we have used. And this particular technology relies on two sets of primers, one with each allele. So you want primers that will amplify both alleles, and they only distinguish they are only distinguished with a single base. And the kind of results that you get are like this. If you have homozygous lines, you get clusters when you look at the call on one allele or another, and on a population you have homozygous and heterozygous lines, and that's what you want to do at the end of the day with KASP genotyping. And this is the general idea, but when you look on diploid species, it can be kind of straightforward because you only need to design a left primer, a right primer, and then you have a product to amplify. Of course, you need to check that the primers are reasonably okay, that they don't have many repetitions, so they don't stick to themselves. But in general, you only need to know the sequence that you want to amplify, and that's it. The problem on wheat is it's a polyploid, and in case that you want to amplify, let's say, the A genome or the B genome, you can get amplification from 2 or the 3 genomes. So you really don't know if your SNP is diagnostic or not, or if you're just amplifying all the genomes. And it looks like this. Let's suppose that you have this SNP here, and the common primer, if you don't think about it, it can amplify the 3 genomes. So you could get that this may be just a variation between the different genomes, but it will look like an heterozygous marker. So we want to have these common primers in places that can be diagnostic to the genome that you are looking at. In general, or the steps that I do inside of PolyMarker is, first you get a list of SNPs, and from them you generate a template sequence, and that has the SNP and the flanking regions. You've lost it to your reference genome. You find all the homologous regions. You make a local alignment, and then you validate it with primer 3. But how does that look? You start with a list of SNPs, and ideally you want to know on which on which chromosome they are. You have a name, and you have to show what is a SNP. So that's all the information that you use to start. And you can generate this file very easily using Excel, and just make sure to save it as a CSV. And that's all what you need. The idea is to have the process as simple as possible. This SNP is converted to a sequence with the SNPs, and you can see that here is an SNP. And the first thing to do is to look at it across all the genome, and so you can get all the copies that you have. So you fish that information out, and you can do a local alignment that includes the SNP. So then you can see what are the differences between the chromosome A, B, and D. And when you look at the local alignment, you can see already there are some differences. The first thing that you want to check is that the SNP, it's actually a SNP, because it can be, like in this case, where there is another possible SNP here between the two markers that we are checking or selecting. But you can see that this SNP, it's not likely to be a real SNP. It's more likely to be a regular variation between the three genomes. So one of the things that the PolyMarker does is to find out these cases. So you know that if you have problems in your analysis downstream, it may be because of this. It doesn't mean that the marker doesn't work always, but it can give you an indication. So usually we discard these cases. And then the next thing to look is to find which differences you have. In this case, we have this T here, but because we are looking at markers of against the chromosomes 5V, we think that this T here, we call it as a semi-specific variation between the varieties. Because if you want to amplify B, your template will amplify B and A. Sorry, we're in the example we're looking at chromosome 5A. In this case, because we have T here, that if we want to amplify to T, we can make a template starting here that will only amplify the reference on, or will only amplify the chromosome 5A. So that's how we get a candidate for KASP markers. And the output that you get is also an Excel file, where you can look at all these details that I mentioned, like the primer type, if it's specific or not specific or semi-specific. So you can, if you have several primer candidates, you can decide which ones you want to actually bring forward for the downstream analysis. To make this more accessible, we have now a website, PolyMarker.info, that where you can send your SNP sequences to design primers. Originally, we started only with Chinese Spring for wheat, but since this tool since this tool came online, people have asked us to include other assemblies. For example, right now we also have the UK assemblies, and we also have the tetraploid wheat for people working on tetraploid wheat as well, and we also have other species that different communities have asked us, and we are happy to include other references as they come. So now I'm gonna talk about other uses of PolyMarker. As I said, it started originally for SNP markers, but since I have all the pipeline already, whenever I need to design other things that or other markers for other types of analysis, I tend to come back to add new variations of PolyMarker. So one of the recent cases is that we have a large population of paragon gamma radiation deletion lines, and this is useful to find lines that have a set of genes missing, so it can help you to make a functional analysis when you use these lines. So in the uniqueness, there is this collection of almost 2,000 gamma radiation lines, but we have sequenced only for 600 of them. We sequenced them with a very low coverage, 0.3x. So we wanted to know if this was enough to validate the deletions, and we are using large window sizes to make up for the low coverage. So we can do this analysis, and this is a collaboration with the Polymarker lab also in the Genome Center. And the original data that we get, it's a lot of when we normalize it, a lot of coverage variation, which is fine, but we get these regions with almost zero coverage, and we want to know if these are real deletions or not. And we have this for the whole chromosome. So we have large deletions and small deletions. And of course, the problem here is does these deletions are real, or is this just an artifact of the sequencing? But anyway, we have these lines available for people to order in the with the deletions website. But yeah, as I was saying, we want to confirm that these deletions are real. So we know that we can detect the SNPs already, and in this case, the example it's on a tetraploid for simplicity, but the principle applies as well for hexaploid. So we know that if we make a KASP primer or KASP marker as we have them before, this will look like as an heterozygous. So the wild type will always look heterozygous. But when you have a sequence missing, or you have a deletion, if you run the same marker, the mutant will look as an homozygous. So in this case, if there is a deletion on the A genome, it will look like a mutant on the B genome. So using the same ideas of getting the sequence and the local alignment, in this case, if we want to validate this deletion on the A genome, we get the same kind of alignment, but we are not looking for a particular position of SNPs. We just look at all the SNPs in this region and which one are specific to the genome that we want. So in this example, we have a case where the SNP can be A specific, B specific, or D specific. So we can actually generate markers for deletions on the three genomes. But in this case, we get this primer pair that is one of the sequences is A specific and here, and the common primer it's B specific, so in D specific. So in this case, we will only amplify these two genomes. So we actually managed to test the deletions this way, and so it's not the typical KASP interpretation. We still managed to confirm that the deletions are real. And well, the other common task is like, can we use this to develop amplicon primers or regular PCR primers? Well, the answer is yes. It's a very similar principle. So again, we start with the idea that we just want to amplify a single sequence, and in this case, we want to get only one of the genomes. So in this case, the same idea of the local alignment. In this case, we're going to go for amplifications of only the B genome. And you can see that there is this specific these two SNPs that are specific to the B genome. So what we get is we use these two SNPs as the primers to amplify. So in this case, PolyMarker doesn't give you a single pair of markers, but a set of multiple pairs that then you can decide which one you want to bring forward for your analysis. So what are the applications that we have already of PolyMarker? So one of them is that, another of the experiments that we have done in the lab over the years, and it's a project that it's always coming back. It's the T-link mutant. So we have two large populations of T-link mutants on Tetraploid and Hexaploid wheat, and right now the data it's available on ensemble plants. So again, it's you can use these lines to look for genes that have mutations that knock them down. So you can use to test if they actually correspond to a phenotype that you expect. So now we have partnered with Ensemble, and they host all these SNPs, and as part of the data that we make public via Ensemble, it's also the results of PolyMarker. So you also know if the SNP can yield to a chromosome-specific primer, and if the SNP is really non-homologous, that means that it's a real SNP and not a difference between the 3 chromosomes. And we also give the set of primers already calculated, so you don't need to go back and run PolyMarker for the mutants that we have identified. We have run it already for all the mutations that are available. And likewise, we ran PolyMarker on all the markers on the iSelect 80,000 and the Axiom 820,000 SNPs chips. And again, you don't need to go and run them again in PolyMarker. All these SNPs are available for download in SerialsDB. And if you have your own reference, you can set it up and run it locally. So the web interface that we have available only it's used for the SNP markers, but we have the source code and instructions to run them locally, and instructions to build your own web server if you don't want to use the online service, or if you want to use your own reference. So in general, PolyMarker it's quite stable, but we do see that there are some improvements that we want to have. Some of them is to have more tests of the amplicon and the deletion marker. So we know that the method works because we have used them internally on the lab, but they haven't been tested intensively outside, mostly because it's not on the website. So we want to make these versions of PolyMarker also available online. And we also want to integrate the new developments on the with pan genome to add extra validations and to use PolyMarker on all the different references. And we are open to other needs the community will have. And finally, I'd like to acknowledge mainly the Huawei group that we worked a lot to get this done, but also on the part of the deletions, the Polnix lab and Hales that has done a lot of the validation with Nikolai Adamski about the deletions, and Anil Thanki on Erlam that has done the visualization for the deletions, where you can see those examples. Koinamati in Ensemble that has helped us setting all the data there, and Mark Enfield in SerialsDB to host the markers. Thank you very much. I'm open for questions now. Hello.
Yes, Ricardo, sorry, I'm having a computer malfunction. I can't seem to get my screen, the questions on the screen and unmute my phone. All right, here we go. So we have a number of questions. First question we had was, which generation of irradiated seeds of Paragon were sequenced?
Sorry?
Which generation of irradiated seeds?
They are M4s. So it's the fourth generation, 3 and 4 generation, depending on the seed.
All right.
So yes, we expect to have some heterozygous mutations, but we are looking mostly to the homozygous mutations.
All right. So what are Hemisnips?
So Hemisnips, it's I don't like that they are called Hemisnips because they are not really SNPs, because the SNP needs to be in a population, but usually people refer to Hemisnips to the different variations between the 3 different genomes. So can you see my screen?
Yes, we can.
Okay, so in this particular example, the ones that are marked as V specific or D specific, these are Hemisnips because here, if you look at it, yes, there is a variation that in here it becomes G and then A, but it's not something that you will find on the population, or probably it can also happen, but these are just variations within the same organism. So that's what it's usually called a hemisnip.
All right. While you're on that page, what does the capital and small letters in nucleotide sequence signify?
What is the what? Sorry.
What does the capital and small letters signify?
So yeah, so in this case, I'm marking with the capital our target sequence. So here, because we want to have the V genome amplified, I mark here on the mask the A as an uppercase, so I know it's my target sequence. And here it helps to visualize where the variations are. So the lowercase are other variations between the genomes that are not specific to our target. So if we were amplifying the let's say well, this should be capital G. But if we're amplifying the A genome, this will be a capital T.
Okay, great. Is only one base difference enough to make it a subgenome specific?
In the case of KASP, yes. Something that I didn't mention is that on the KASP technology, it's designed to be very sensitive to the first base of the primer. So that's why it is here. Of course, if you have two SNPs one after the other, it will be even more sensitive. But in general, with KASP, and again, this depends on the polymerase that you're using. In the case of KASP, if you have variations later on the primer, they don't affect the amplification that much. So yes, it's something to take in account when designing markers or primers for polyploids.
So what does the input look like for a deletion?
So the input that we use for the deletion, it's the sequence of the region that you want to design markers for, and if you to let know the program which is your marker, instead of having regular names, the title or the name of the sequence should be corresponding to the output of SAMTools FAIDX, which gives you the chromosome name and the region that you're amplifying. So PolyMarker uses that information to know where it needs to match.
Okay. So what's the error percentage for genome specificity of the primers designed with PolyMarker?
That's a very good question. So in general, when we did this analysis, we had like roughly 80 to 85% of the markers work as expected. But that was done with the old version of the assembly. They went from 2014. I don't know if it. I haven't run these numbers. Well, I haven't done a large scale experiment again to see how much it has improved, having a better reference.
All right. Is PolyMarker useful for designing PCR primers for diploids like rice?
Yes, yes, it is. So if you look at the list of species that we have here, we have several diploid species. So we could add a reference of rice if the community wants this added. They just need to email us. It doesn't take that much to add it to here. But if you again you can do it. You can run it locally as well. So it yeah. And another thing that PolyMarker does that I didn't mention is that if you have multiple copies of a gene, it will try to figure out if or to make it genome specific even within the copy. So let's say you have a duplication on one of the chromosomes, so you have one for one genome, two for the B genome, and only one for the D genome. It will make them specific to the copy in the genome that you are targeting. So if you have this kind of duplications in rice, as long as they are on different chromosomes, it should make them genome specific. And even in the case that they are that it cannot distinguish which of the copy is, at least it will give you an indication of how reliable the marker is.
Yeah. So just to follow on a little bit about that in terms of how easy it is to add assemblies, is it a long process or is it relatively easy?
And if the names of the reference chromosomes are kind of standard, having the chromosome and the chromosome group as the first two characters in the reference, it's kind of straightforward. The only thing you need to do is to make a blast database, and that's it. If the names of the chromosomes, or if you have scaffolds and you need a different way to identify from which chromosome it is coming, it may take a bit longer, because on PolyMarker you can define rules of how it knows which is the chromosome, but you need to fiddle with the code to do that. But in general, it's not difficult.
So as you know, we'll be releasing IWGSC RefSec version 2.1 soon. Will you add that? Because it might be very useful for seeing the differences between the two assemblies.
Yes, definitely. As soon as it is public, I'm happy to add it to the website.
All right, great. Now the next question is, if the reads for each chromosome are completely equal, can you still use the pipeline?
You can. It will tell you that they are not specific, but that's a problem that you will have with the primers anyway, because you cannot really, if you cannot distinguish them visually or by yourself, also PolyMarker will not be able to distinguish them. For those cases, or for those complex cases, we suggest to look for SNPs on the promoter region that tends to change more, and use that to genotype.
All right. For some markers, you can have info of linked SNPs that are not necessarily semi-specific. Can PolyMarker take this information into account and avoid designing primers on these positions?
No. We don't have that directly on PolyMarker.
Okay. How long can a primer be before adding the tail?
So we use them between the shortest. It will be 19 bases, but ideally it should be around 21 bases. And I think we have used some up to 26 bases, and still fine.
Okay. One of our questions is that they've seen better results with KASP created with Cracon from LGC as compared to the ones created through PolyMarker. Do you have any idea why? As you would expect, the PolyMarker generated KASP should be better since those are derived from the reference genomes.
Probably because they know better the chemistry and I don't know how they work internally, but as we don't take in account any of the information of how they produce the markers, I cannot compare it directly, but my assumption will be that. And on the top of that, if they ensure that it will work, if you get the service, I guess they will test them before releasing them. But yeah, I don't know what other difference will be.
The next one is, are the seeds of Paragon deletion lines available publicly?
Yes, they are. So if you go to the with the deletions website that I mentioned, this one, there is a link there to order the seeds from Seed Store in the Uniqueness Center.
All right. Is there any capability for users to leave comments on the success or failure of a particular marker, or to share successful PCR-KASP conditions?
We don't have that on the website. I see the point of sharing that information, but I don't have the website set up for this.
Is that something that could be done, or at least give have a way that they can?
So for the markers that we have already designed, probably it's something that could be done, but if it is just if it is for the primers that people are submitting to the website, as the assumption is that only the user gets their own primers, it's not something that we plan to add.
Okay. Does PolyMarker have a function to help design homeeolog specific primers optimized for RT-qPCR?
So yes and no. So it's a bit technical. So PolyMarker will search for the positions that you can use to be homolog specific or specific to the genome that you want to use. But since that's not a technology that I have personal experience, I don't have a setup of what to give to primer 3 to validate the dataset. However, you can give to PolyMarker a preference file of primer 3, and it will force primer 3 to use those parameters if you know how to set it up for certain technology.
All right. I'm not sure you'll be able to do this one, but is it possible to run an example right now on the website? For you to literally open it up and actually do it. Is that possible?
It should be. I haven't checked that it is working right now. But
if it is not, we could always do it as a separate video and add it on. I think it's a great idea.
Example here, and it should run. So now it is running, and it will take probably 1 or 2 minutes, and here it is. Yep, it is working. And then the other thing that we have here on the website is that you can look at the local alignment of the markers. So here are the markers, and you can see how it looks. So if you have more sequences, in a highly repetitive region, you can also have a look at it. And you can also download the FASTA file with these alignments in case your marker doesn't work and you want to try a different pair. You can also use that information. And you can also download these 2 buttons. The primer gets an Excel file of this, and the mask will get this file. And here are the upper and lower cases that we were displaying before.
All right. So is it just curiosity, is it possible to do a training seminar, or would that be something that you could do on showing people how to? I'm not sure how it would work, but given that it would be virtual, but
yeah,
I wonder if it would be possible to show people how to actually do it live. I mean, to have a session where you can share screens back and forth.
If there is interest, yes, and I mean, we could follow up on how to run it with your own references if we have enough interest. Probably something we could do.
So we'll add that to the survey of all the participants and see if there's an interest in doing that, and then can see if it's at all feasible, given that it would be virtual. Okay. The next question is, I did not get the point how to design KASP primers for the detection of deletions. Do you have a scheme of how to do that, and have you tried it in practice? That this particular person tried to do it, but they did not work. Okay,
so we did try it, and we do know it works. Unfortunately, I don't have in hand the examples. So this is the figure in the case of KASP. What we have to do is to make sure that when okay, because we have a population where we assume that some of them have the deletion and some of them will not, or most of them will not. instead of running just the marker on a singleton, you need the context of how they run across all of them. So in this case, we are amplifying the A genome and the D genome, and if the A genome is deleted, we expect to see mostly a cluster of heterozygous SNPs, and just if we only have one sample that has the deletion, a single point that it's an outlier. If you're looking at the full population where we have more than a single line with the deletion, you can also look at 2 or 3 lines, and so you have a small cluster, 2 or 3 lines that will be homozygous. But yes, we have used it, and it has worked for us.
All right. Can the same website be used for designing KASP markers based on indels?
No, we haven't think about how to use indels in PolyMarker. We do see the value of that. I mean, if it is for small indels, and if it is for these deletions, large deletions like the examples that we have here, we are looking at getting some funding to get this developed.
Can PolyMarker increase the amplicon size if the primer criteria are not met?
In the case, yes, in the case of not on the website, but on the command line version, there is an option where you set what is the size of products that you want to produce, and it will go as far as you tell it. So we have tried it up to, I think, 200 base pairs. We don't have it tested as much as the small product sizes, but yes, it can. And for the amplicon, it will. For the amplicon version, if you have a long sequence, it will give you all the possible pairs that are genome specific on that range. So it's not that limited. I think it has a default of 200 base pairs product, but I think it can go further. Again, because these are not used, or we haven't used this as heavily. I don't have like exact numbers, but yes, it can be done.
So how difficult is it to do that? Can someone without specialized IT training do it?
So in principle, you just need to install it in your computer. It's a command that you can run on the command line. So if you have basic bioinformatics knowledge, it should be straightforward. It is documented on the website, or I'm finishing the documentation on the website and adding some examples there. But of course, I'm biased. I say it simply because I did it. So, but I try to make it as easy to install as possible.
All right. So here's the next question. How many bases should be genome specific in primers for RT-qPCR looking at gene homeolog expression?
I don't know. Again, I haven't personally worked with RT-qPCR for expression, so I don't have an answer for that. Sorry.
Okay. One of the next suggestions, instead of a question, is to add the soybean genome to the website.
Yes. So if they send an email with the with the reference we on the next update we can add it.
All right. How do you get the alignment on the website?
You just click here on the mask, and sorry, and you download it, and then it's just a FASTA file that you can open with your favorite viewer. So here it's the alignment for the tree marker. So it's the same information on the website, but here you can do more analysis if you want.
So can you provide an example on your website of how to set the input, or how to do the setup for the input of the deletion and insertion?
Okay. So again, on the website we only have the KASP markers, but on the other web, I don't know how to use PowerPoint. on the website with the reference with PolyMarker in PolyMarker, or I'll do it on the website. So here in PolyMarker.info you have in the about, it takes you to the GitHub, and here on the wiki again that I'm writing right now, but there are examples here of how should be the input for different things. So I'm finishing the documentation for the other versions of PolyMarker, and on the website itself for the KASP markers, if you click here on example, that's what I just did. It gives you these are the examples that I just run. So you can copy and paste it to get an idea.
So we just had a question regarding what you were doing. So what was the alignment viewer that you just used to view the downloaded file?
This one, it's called AliView. AliView, it's here.
Okay. All right. Can you use the primer for homeolog specific expression analysis? I think you kind of addressed that a little already.
Yes.
Elaborate on that a little bit.
Yes. I expect that it should work. The version for amplicons, it should work. But I haven't tested personally. So if you try it and it works, I will be happy to get any feedback or tricks to improve it.
All right. I think that covered most of the questions. So if anyone has any questions or wants to provide input, how do they reach you? Can you put your contact information back up?
Yeah, so it should be the last slide. Yes, so the best way for PolyMarker related issues is if you go to the GitHub website, you can add a new issue, or you can write me an email that I forgot to put my email here.
That's okay.
But
we can put that in the survey.
Yeah, we can put it there. And I'm super easy to find with this super long name that I have. You just write Ricardo Ramirez-Gonzalez and you find it.
Yeah. There have been questions. Are you planning on doing trainings of using any other tools that would be useful for scientists or for breeders that you're working on?
So not actively, but we are open to that. Like, unfortunately, this year has been very very bad for organizing things live. But yes, we are open to that, to do training. Yeah.
So we'll definitely put the question on the survey and see what people would have an interest in doing. I think somebody raised their hand. Okay, no, I guess not. All right. So with that, I want to thank you again, Ricardo, for a great presentation of a very interesting tool. I think that will be very useful to a lot of individuals, and we will, for the community, we'll explore maybe setting up some kind of a training session for using it. I want to thank everyone for attending or watching the video after it's posted on the YouTube channel, and encourage you to join us for our next webinar on the 19th of November, decoding the polyploid wheat genome using gene networks. So look forward to seeing everyone then, and thank you again for joining us today.